Namaskar dear students, welcome you to our channel by Gulab Sir. As a part of series of lectures, we are going to learn today about meiosis. We already have discussed about mitosis. Now let us continue with meiosis. As you know that meiosis is a reduction division and it occurs in germinating cell, that is called generative cells, which are supposed to produce the gametes and spores. Meiosis occur in cells called myocytes, undergo reduction division to reduce the number of chromosomes to their half. That is what diploid cell become haploid. How it happens? For that I have made one format. Although it is a long process, it required more time to understand in meiosis 1 and meiosis 2, these two subphases. But I tried with this summarized form to understand at a glance. Let us have a look. Here, a cell division in which that two new daughter cells are produced, which has, or four new daughter cells are produced, which has half the number of chromosomes to the parental cells. See here, two sets of chromosomes. After interphase, that is with growth, it becomes enlarged in size with prominent nucleus and cytoplasm. Undergo meiosis 1 where reduce the number of chromosomes to its half. And later second meiosis is mitotic. Therefore 4 cells are produced with same number of chromosomes to the cells present after meiosis 1. So parental cell has 2M and gametes which produce or spores which produce, they have haploid set of chromosomes. But how it happened that 4 cells they received haploid set of chromosomes? Initially, the cell which undergo meiosis has, suppose one cell is equal to 23, one chromosome is equal to 23, just have a scale. Thus, these two sets chromosome equal to 46 chromosome. Now here 46 and each cell receives 23. That means half to the number of chromosomes present in the parental cells. Now these cells supposed to undergo meiosis. Before that, it undergo growth phase. That already we discussed here. That interphase, cell enlarge in size, where during synthesis phase, because during growth phase, G1 synthesis and G2 phase, during this synthesis phase, replication of DNA occur. That means monovalent chromosome become double, that is bivalent chromosome. That is very important because increasing number of or amount of that nuclear material equally is required for the meiosis. Now these cells undergo meiosis 1. What is meiosis 1? Movement of homologous chromosome. That one of the member of homologous chromosome move to one pole, another to another pole, forming two daughter cells with haploid set of chromosome. So here it was 1, that is 2 chromosome. Here it is 1 chromosome. Here it was monovalent. Here it is bivalent. This is the only difference. Otherwise, each cell receives only 1 chromosome. Therefore, this is haploid to the parental cell. Therefore, this first meiosis is called meiotic and it is a reduction division, reduces to half. Now these chromosomes, these cells, these two cells produced from this myocyte undergo meiosis 2 where splitting of chromosome occurs. This bivalent chromosome split into 2 monovalent. Thus each new daughter cell receives 1 chromosome. Here also this bivalent chromosome split into 2 monovalent. Therefore 2 newly produced daughter cells receives 1 chromosome. And thus all 4 cells are produced from these very parental cells having haploid set of chromosome, as there was 2 chromosome here, 1, and that is what we have to understand throughout the process of meiosis. So this makes you very easy to understand what is going on through meiosis. And now we have this haploid spores either in gametophytic phase of bryophyte and pteridophyte, or these are the gametes in animal where that cell myocyte undergo meiosis or gametogenesis to produce the 4 gametes. Now let us understand this entire process of meiosis, how it occurs. See here I have written that meiosis occurs in 2 subphases, meiosis 1 and meiosis 2. Meiosis 1 is very long, since in meiosis 1, prophase 1 has 5 subphases, that is liptotin, zygotin, pacatin, diplotin and diakinesis. That also we have to understand, including metaphase 1, anaphase 1, telophase 1. Here meiosis 2 will be continued. After that telophase 1, meiosis 2 will have prophase 2, metaphase 2, anaphase 2 and telophase 2. Let us understand in a sequence what are these phases. Before that, I have this diagram to explain you exactly what is meiosis. Otherwise, in next, you can see their notes are given and that you can refer. But before that, to understand meiosis, we must go through this diagram so that what is happening with that cell. Let us see, this is the first phase of the prophase leptotin. Now prophase begins after interphase, that is what metabolic stage. So in prophase, nuclear membrane, nucleolus and organelles are intact. You see all these stages, leptotin, zygotin, packaging, diplotin and dikinesis. That nuclear membrane is there. Prophase 1 or prophase has nuclear membrane, nucleolus, organelles. Centrioles are in the vicinity of nucleus. So cells is getting ready for cell division. So in meiosis 1, prophase stage has first subphase leptotin where nuclear membrane, nucleolus, all organelles are there and chromosomes are long cylindrical, thread-like. But they are beginning to appear as a bivalent chromosome. In the late prophase, that nuclear membrane, nucleolus, organelles, all they get disappear in the late prophase and enter into metaphase. But there are subphases. What are that? After leptotin, zygotin, where pairing of chromosome, that is called synapsis. See, here these homologous chromosomes come close to them. Like that, other 22 pair of homologous chromosomes in human being come together and form the pairing of homologous chromosomes. That is called synapse. See, synapsis I have written here as a notes in prophase 1, leptotin. See, prophase 1, leptotin and here zygotin. It is very clearly written in zygotin, pairing of homologous chromosome called synapsis that form the synaptimal complex. This is pairing of chromosome. Next, that is packaging where crossing over occur. What is crossing over? Exchange of segment between this non-sister chromatid of homologous chromosome. And here it is clearly seen that how segments are exchanged. With that capital A, small b has gone. With small a, capital B is come, and that is the new combination. That is called recombination, which occur during packaging stage. See in diagram how packaging stage is shown. See here, packaging stage, crossing over occur. After packaging stage, diplotin where chiasmata formation. This cross what I am showing here, this is called chiasmata. That is what still it is attached physically. Functionally it is completed, segments are exchanged, but still it is attached. That attachment point is called chiasmata. Chiasmata formation in diplotin and see in dikinesis where terminalization begins. That crossing over is completed, chiasmata start moving from center to the periphery and thereby from inner to outward, that movement of that bivalent chromosome to separate. And thus that two chromosomes with new combinations are produced. Now that cell enter into metaphase 1 where nuclear membrane, nucleolus, all they are disappear. See, although I have not drawn their nucleolus, but we have to consider that nucleolus is there at every stage because till the metaphase stage nucleolus is intact. But when metaphase stage come, that nucleolus also disappear. Nuclear membrane disappear, organelles disappear, centriole split and move to the opposite pole and form the spindle fiber. See, these are the spindle fiber. As well as chromosome gather in the equator. That is what pair chromosome. 23 pair of homologous chromosome gather in a pair at the equator where spindle fiber attached to their centromere. Now after metaphase 1, anaphase 1 where spindle fiber begin to contract. Since spindle fiber begin to contract, that chromosome begins to move or drag towards the polar region. And thereby this chromosome, that homologous pair of chromosome, one of the homologous chromosome move to one pole, another move to another pole. And since they move to the opposite pole, now each pole receive haploid set of chromosome. Therefore, two new daughter cells are produced from this parental cell with haploid set of chromosome. See here there were two chromosomes. Here each one has one chromosome. Now here it is clear that at the end of meiosis 1, two haploid daughter cells are produced. And that is what we already learned in that format. See what I did explain to you here. That meiosis 1, that two cells with haploid number of chromosomes are produced. And this is what we learn meiosis 1 with leptotin, zygotin, packaging, diplotin, dikinesis, metaphase 1, anaphase 1 and telophase 1. At the end of telophase 1, these are the two cells which are produced, are already shown here also with diagram. I hope you did understand what exactly that prophase 1, metaphase 1, anaphase 1 and telophase 1. So you can go through this diagram for detail again and again to get it clarified what is being explained. So after telophase 1, now cell enter into the meiosis 2. But the phase between this meiosis 1 and meiosis 2 is called interkinesis. That means there is no any growth, there is no interphase. But immediately this telophase 1 stage get enter into prophase 2 of meiosis 2. And that is what we have to understand as the next stage. See, meiosis 2 is a mitotic where number of chromosomes remain same. And therefore, you see here the number of chromosomes is 1. Therefore, at the last also, each cell will have 1 chromosome. That is what meiosis 2 is, mitotic, where the number of chromosomes remain same. So, meiosis 2 begins with, again, prophase 2. See, in prophase 2, as prophase 1, cell membrane is intact, nuclear membrane is intact, nucleolus is there, all other organelles are there. Immediately after prophase 2 or at the end of the prophase 2, nuclear membrane disappear, nucleolus disappear, organelles disappear, and centriole split to opposite pole. But here the cell division in meiosis 2 is right angle to the cell division in meiosis 1. That is very important point. There it was like this, here it is like this. In meiosis 1, division what? In this direction. Now here meiosis 2, division is in this direction. That means meiosis 2 arrangement of spindle fiber is right angle to the spindle fiber present in meiosis 1. That is important point we know as a NEET. So here this chromosome again get into the center of the cell. That is called again equatorial plate, where spindle fiber attached to the centromere and that two gonadotropes that newly produce plates over the centromere. So spindle fiber attached to the kinetochores. Now this is a metaphase stage where nuclear membrane disappear, nucleolus disappear, all organelles are disappear, spindle fibers are formed, chromosomes are at the thickest and they are gathered in the center and attached with centromere. This is what the explanation about metaphase 2. Now what is anaphase 2? Continue. As that condition, all conditions which was metaphase 2, same condition in anaphase 2 except spindle fiber begins to contract. Here spindle fibers are there, but spindle fiber begin to contract. As spindle fiber begin to contract, that spindle fiber attached to kinetochore drag behind the spindle fiber. Thereby, bivalent chromosome, see, 2 arm on either side, here 1 arm on either side. So bivalent chromosome turn into 2 monovalent chromosome because of contraction of spindle fiber. So that centromere split, kinetochore along with spindle fiber carry that chromosome, monovalent chromosome, to the respective pole in say both the cells. So because these two cells were produced as a product of meiosis 1, now both the cells undergo meiosis 2, therefore same condition will happen in every stage in both the cells, and therefore here also movement of that chromosome with the contraction of spindle fiber, thus these bivalent chromosomes split into 2. Now as we know that during Pachydian stage crossing over occur, and therefore here these cells what we are going to produce will have 4 new combinations. As you see this capital AB, capital A, small b, small a, capital B, small a, small b. Now in telophase, these chromosomes reach to the opposite pole where nuclear membrane reappear, nucleolus reappear, organelles appear, and this chromosome will be with this gamete having capital AB combination. Another chromosome will be with another cell, that is another gamete with capital A, small b. This again out of this will be with this cell having small a, capital B, organelles, nuclear membrane, nucleolus there, and gamete is formed. So 4th cell with this combination small a, small b. So 4 gametes are produced with 4 type of combination, capital AB, capital A, small b, small a, capital B, and small a, small b. Thus 4 types of gametes, haploid cells, gametes are produced from very diploid cell of meiosis which was undergo meiosis 1. Thus here meiosis is completed. I hope you will now understand stepwise how it has happened. So 4 gametes are produced that together, they remain together in plant called tetrad, and they get separated and the pollen grain formation begins. But in human or animals, these undergo spermiogenesis, that means formation of spermatozoa, and later spermatozoa undergo spermiation, that is release of spermatozoa. So these cells, although they look circular, but later they differentiate to form the sperm having acrosome, nucleus, middle piece, and tail, that is called spermiogenesis. So that process we are not supposed to understand. We have to understand only how 4 different types of gametes are produced by the process of meiosis. I hope you did understand. For entire explanation, stepwise explanation, please go through these notes which are written later. See here, this is about meiosis 1. In meiosis 1, prophase 1 is the first stage written about prophase 1. Prophase 1 includes liptotin, zygotin, pacatin, diplotin and dikinesis. That is also explained. Second, meiosis 2, metaphase 1, metaphase 2, anaphase 1 and telophase 1, as well as meiosis 2, which has prophase 2, anaphase 2, telophase 2, and thus the meiosis is completed. So here these notes are available. You can go through these notes as a point to be mentioned under each heading, supposed to write as a part of that notes. I hope you did understand the entire process of meiosis, how it is being completed in these two phases. For detail, you would have to see this video again and again. If you see 2-3 times, definitely you are going to understand every and each with this clear-cut diagram explanation. I hope this is very clear to you to understand meiosis. And हमें ये पता है कि आप हमारी वीडियो टाइम टू टाइम देखते हैं और आपको बहुत अच्छे भी लगते हैं, लेकिन हो सकता है इस स्टडी के फेज में आप भूल जाते हो, इसलिए मैं आपको अपील करता हूँ कि शेयर करना और सब्सक्राइब करना नहीं भूलना, लाइक करना नहीं भूलना, ताकि आपको हमारे नए वीडियो देखने के लिए मिलेंगे. सो ये हमारे लिए एक इंस्पिरेशन रहेगा. अगर आप लाइक करेंगे, शेयर करेंगे और सब्सक्राइब करेंगे तो हमारे लिए एक मोटिवेशन रहेगा और अच्छे वीडियो बनाएंगे और क्लियरली आपको एक्सप्लेन करने का प्रयास करेंगे. थैंक यू. हैप्पी लर्निंग.